Hello everybody, welcome to Ms. Mugwara online video lecture, and today we're covering gene expression and epigenetics. So let's go ahead and begin. Learning outcomes for this chapter. After studying this chapter, you will be able to define epigenetic, explain how small molecules binding to histone proteins control gene expression by remodeling chromatin, explain how microRNAs and microproteins control transcription, and discuss how viral DNA, non-coding RNAs, and repeated DNA sequences account for large portions of the human genome. Genome is like an orchestra, just not all of the musical instruments play with the same intensity at every moment. Not all genes are exposed continually at the same levels. So just think about it. You made of a billion of cells. Every cell in your body has exactly the same DNA compositions. So your chromosomes, your DNA, your genes are the same in every single cell. So then how it is possible that your cells are so different? Why a cell of your pancreas, for example, express the genes means they use these genes to make insulin, and cells of your skin do not use those genes. Even those genes for insulin production are present in every cell of your body, in your neurons, in your skin cells, in your liver cells, but you only use these genes for insulin synthesis and secretion in cells of your pancreas, right? So how it is possible? Well, that's what it says, that not all the genes always expressed. Right, so at the same time. So that's what this statement tells you. Well, some genes, however, are always expressed in all cells, and we call them housekeeping genes. Other genes are more specialized, like the example that I gave you about genes for insulin synthesis. They're very specialized, and they became active as a cell differentiate or challenges arise. Right, so epigenetic changes. Now, epigenetics refers to inheritable changes in your DNA that don't change actual DNA sequence. So we know what mutation is. We already talk about mutation. When we change the sequence of DNA, when you change DNA sequence, then you alter the production and synthesis of proteins, and that will affect function of the cells. But what if we have changes that do not change DNA? However, they still inheritable and they still affect your functions, right? That's what epigenetic is. And that means these changes are potentially reversible. Changes to the chemical groups that associated with DNA. So it's not change to DNA, it's change to other chemical molecules that associated to your DNA that are transmitted to daughter cells after cell division. This is what, again, epigenetic is. So our bodies are a bunch of parts that build from different kind of tissue, from nervous tissue, skeletal muscles, where there's muscles over here, muscles, connective tissue, your bones, connective tissue, blood, epithelial tissue that is part of your skin. Those parts are made of different cells, right? Like your neuron, like your muscle cells, like your epithelial cells. And those cells have unique epigenetic marks on their DNA, right? So even the DNA might be, as the DNA is the same, right? You have the same 46 chromosomes, you have the same DNA, but those unique marks, epigenetic marks, are different in different type of cells. So just kind of to summarize it, epigenetics is a study of these chemicals that are related to DNA and that actually changes how DNA is expressed. Those changes can be passed from generation to generation, and those changes does not alter actual DNA sequence. So here's example, pancreas. Pancreas is a dual gland. It has exocrine and endocrine part. So pancreas is a gland made of cells, right? So you have cells, make an organ. But some of the cells, they release digestive enzymes into ducts. So they part of endocrine system of pancreas. And other cells, they secrete hormones directly into bloodstream. So how it is possible that different cells within the same organ that have the same genes, but they perform different functions? That actually what produces these endocrine or exocrine cells is a differential gene expression. So if a transcription factor is activated, some progenitor cells follow the exocrine pathway, and other progenitor cells respond to a different signals and yield daughter cells that follow endocrine pathway. So the next diagram shows us this process. So here we're building up a pancreas made from stem cells. So remember, when stem cells divide, they produce another stem cells and progenitor cell. So here we have exocrine and endocrine progenitor cells that will follow different pathway of differentiation if different factors are activated. So it can become exocrine progenitor cells and then mature into exocrine cell called acinar cells. Those cells will secrete digestive enzymes, or the same progenitor cells, depends on what genes are activated, become endocrine progenitor cells and can be developed further into beta cells, alpha cells, gamma cells, or F cells. And all the cells will have different functions. Right, so if you go back, that's what it says. If a transcription factor is activated, some progenitor cells follow exocrine pathway, and other, if different signals are activated, will follow the endocrine pathway. Right, so the point we're trying to make over here is that despite the fact that all the cells in your pancreas have the same DNA, some will develop into cells that secrete digestive enzyme and help you to digest your food, and some will develop into cells that secrete hormones like insulin, glucagon, somatotropin. Right? So that's all how we express this DNA. DNA is like a switch. Genes, you can switch them on or switch them off, right? So in these cells, in this cell that is exocrine cells, genes that produce insulin and glucagon are switched off. So they're not working genes. But in a beta and alpha cells, genes that produce digestive enzymes are switched off. And genes that are responsible for insulin or glucagon synthesis are on, so they are active, right? So then how do we control of this gene expression? So again, why do we need this gene expression? Because in your pancreas, you need genes that code for insulin synthesis. You need these genes to be expressed. And in your neurons, for example, you don't need these genes to be expressed because you don't want your neurons start secreting insulin. And where's this insulin going to be? It's going to be, like accumulated in your brain and your spinal cord. So neurons should not use those genes that are responsible for sweat production, for example, right? Because you don't want to produce sweat in your brain. And at the same time, you want in the neurons, you want genes that produce neurotransmitters to be activated, right? So that's what the gene expression is. So how do we regulate which genes will be working and which genes we want to turn off? Well, the protein encoding gene that encode for protein contains some controls over its own expression level. For example, promoter sequence. So remember, for gene to be transcribed, so we have DNA, that's what your gene is made of, DNA. Then we need to make a copy of this DNA, make messenger RNA. And to make this messenger RNA, we're going to start with promoter. So that's a part of DNA molecule where transcription factors will bind, so we can make a copy of a gene. So we might have mutation in this promoter sequence that either increase the expression of this gene, so you will make more proteins, or block the expression, you will make less proteins. Also, sometimes we have extra copies of gene. Now, if you have extra copies of gene, obviously you will produce more proteins that this gene code for, right? That's our control over gene expression that already part of that gene, right? But much of the control of gene expression occurs in two general processes: chromatin remodeling. We can switch on and off the genes by remodeling chromatin, and microRNAs. Proteins also control each other via microproteins. So here we have three types of control: chromatin remodeling, microRNA, and microproteins, right? So let's look at more details. So here your DNA, right? So here's your chromosome. Right, so chromosome is made of chromatin. Chromatin is DNA wrapped around these histones. So we can have histone modification, right? So you see those chemicals that attach to histones. That's what epigenetic is. When you're not affecting the DNA, you're affecting other molecules associated with DNA, right? So you don't make mutation. You just attach chemical to DNA, like this histone modification. So it can, for example, if we modify histone in a way that it package chromatin very tightly, then we cannot express this part of DNA. Or it can be DNA methylation, when we methane group is attached to DNA and it's kind of switch it off. Right, or our microRNA. So we know that we need transcription factor to open up DNA so we can make messenger RNA. So from DNA, from gene, we go to messenger RNA and to proteins. And this is our working protein. But if we have microRNA that will be attached to messenger RNA, it will block the translation of microRNA into protein, so we don't have a protein, and this means we don't express the gene, right? So that's a control of gene expression. Okay, so here one control that we mentioned is DNA methylation. It's an example of one of the many mechanisms of epigenetics. DNA methylation often inhibits the expression of certain genes, right? So we don't want so again, we have genes to make proteins, right? If we inhibit the transcription of these genes, then we don't have proteins, And this is how we turn off a specific gene. Methylation process might stop a tumor-causing gene from turning on, preventing cancer. I guess that's a good news. So here's the DNA, double helix. So here's our two strands. And if we add this methane group to the DNA, as it showed here on the diagram, then it will prevent transcription of the DNA. If we cannot transcribe DNA into messenger RNA, then we cannot make protein. That means this gene is off. So here's another mechanism, chromatin remodeling. Histones play a major role in gene expression. They expose DNA when and where it is to be transcribed and shielded when it is to be silenced. Major types of small molecules that can bind to histone include acetyl group, methyl group, and phosphate group. So again, if we have a DNA, right, and we need to make a copy of a gene, if we attach acetyl and phosphate groups, that we will turn transcription on, right? So here's our transcription off. We attach this group and we turn transcription on. That means gene is working now, right? We transcribe it, we're gonna make protein, hopefully. And also, if we remove this methyl group, it's also turn transcription on. So attach this group, remove this group, and we will turn transcription on. So we will express this gene. Now, we already know that DNA, so here's the shown here in the blue color, that's a DNA molecule, and it's wrapped around histones. And this combination of DNA, histones, and small amount of RNA is called chromatin. Now, in most cell types, only 2% of the chromatin is open and easy to read. So see how this chromatin is very tightly packed around histones, making a nucleosome. So nucleosome is the DNA and histones. And this is hard to read DNA. That's a hidden DNA over here. And this DNA, this gene over here, is easy to read, right? So it's interesting that only 2% of chromatin is easy to read DNA. And now you see how we add those chemical groups to histones. And so you see, so here's the histones are proteins, so they have this made of amino acids. So if we add some chemical groups to amino acid, we can open the DNA and read it. And if we can read it, then we can express it. So here's the example, like acetyl binding can subtly shift histone interaction in a way that eases transcription. So if you look over here, so again, our DNA around histone, and we have this Tata box, and this is the beginning of the translation, right? So when we add this acetyl binding sites, and Tata box is exposed, and transcription factors and RNA polymerase can now bind to it, right? So I'm sorry, I talk about translation. Translation happened next. This is transcription yet, right? So to transcribe DNA and make RNA, we need to attach RNA polymerase to this Tata box, and to do it, we wanna shift the histone. Like, see, in this situation, histone kind of covers this Tata box, but when we attach this acetyl binding sites to histones, we shift it, and now Tata box is open, right? So if Tata box is open, we can turn the transcription on. Right, so so on this diagram, you see again the chromosome, And epigenetic mechanisms are affected by those factors and processes as a development in utero, environmental chemicals, drugs, pharmaceuticals, aging, and diet. And all of these factors, all of these mechanisms affect this epigenetic factor. So they, your diet will affect your DNA. It's not gonna make a mutation to your DNA, but it can add DNA methylation, for example. So you can, if you eat diet that is rich in methyl group, because methyl group is just a chemical. So if you eat food rich in this methyl group, it might be attached to your DNA and turn it off, right? So DNA methylation that we just described, the methyl group, and methyl group is epigenetic factor found in some dietary sources as well, can tag DNA and activate or repress the genes. You can activate genes that you're not supposed to activate, like that might be promote cancer, or you can repress genes that you're not supposed to repress. Histones and proteins around which DNA can bind for compaction and gene regulation can also be affected by those epigenetic factors. So over here we have a gene that is inactive and we cannot express it. Now health endpoints like cancer, autoimmune disease, a mental disorder, diabetes, they also have chemical, and those chemicals are epigenetic factors that can be attached to those histones and modify them. So histone modifications, the binding of epigenetic factors to histones, tails that alter the extent to which DNA is wrapped around histone and the ability of genes in the DNA to be activated. So the point here is where we can alter our phenotype, our functions, if we change the DNA sequence, but it's not epigenetic. Epigenetic when chemicals are attached to DNA or histones and they either make genes easy to be expressed or hard to be expressed. Right, okay, so next one. Another way how we regulate gene expression are microRNAs. They belong to a class of molecules called non-coding RNAs. Non-coding means they do not code for proteins. They just 21 to 22 bases long. Human genome encodes at least 2,555 distinct sequences of microRNA that regulate at least 1/3 of the protein encoding genes. So we have those part of the DNA, right? So part of the DNA that code for microRNA. Those microRNA not gonna be involved in the synthesis of proteins, right? They are non-coding RNA. However, they regulate protein encoding genes. A typical human cell has from 1,000 to 200,000 individual microRNA molecules. When a microRNA binds to a target messenger RNA, it prevents translation. So let's imagine you have, not imagine, that's a fact. You have a gene, right? And you transcribe this gene, you make messenger RNA. Why would you make messenger RNA? Because you want to synthesize protein, right? That is encoded by this messenger RNA. But if microRNA attaches to messenger RNA, we don't have translation. And this is how we regulate the expression of gene, right? So I just want to remind you again. So what we have DNA, right? So from DNA, the purpose of this DNA is actually to make proteins. So first you're going to make copy of a gene. You're going to make messenger RNA. And then from messenger RNA, your cell's gonna use this to make protein, right? So now, some cells, well, some proteins you want to synthesize only inside specific cells, like insulin, that is a protein. You only want this insulin to be synthesized in pancreas, right? Like neurotransmitters, you want them to be synthesized only in your neurons. Some, growth hormone, that is also protein, right? I don't know, actin, myosin, that is proteins of your muscles. So you want specific protein to be synthesized by precise cells. You don't want too much of proteins. You don't want too little, too small amount of protein. So that's need to be regulated, right? So how do you regulate it? Well, one of the ways that's shown over here, for example, you made this messenger RNA, so you are on your way to make proteins, but then you attach microRNA, and you block this production of protein, right? So that's another way to regulate gene expression. MicroRNA controls gene expression mainly by binding with messenger mRNA in a cell cytoplasm instead of being translated quickly into a protein. So instead of mRNA be translated to a protein, the marked mRNA will be either destroyed and its components are recycled, or it will be preserved and translated later. So if the level of particular microRNA is underexpressed, its level in the cell is abnormally low, the protein it normally regulates might be overexpressed, so its level will be unusually high in the cell. If microRNA is overexpressed, so we have too many microRNA, its protein will be underexpressed. Its level will be usually low, unusually low, right? So that's how microRNA regulates the amount of protein that is synthesized by your cells. Practical application of microRNA can be cancer treatment. In cancerous cells, microRNA genes can be damaged by mutation. So remember, you need this microRNA to stop transcription. So you don't want that protein anymore. So how to switch off the synthesis of protein? You're going to attach microRNA to messenger RNA. But what if you don't have microRNA? What if the genes that make microRNA are damaged by mutation? A mutation in a microRNA gene can leave the cell without that particular microRNA or reduce it a low level in the cell. Abnormally low level of microRNA can lead to overexpression of genes. So if you have a very low microRNA level, then you cannot stop the production of protein. Then that means you will have too much protein that is regulated by this microRNA, and it can lead to cancer development and progression. Abnormal expression of microRNA has been implicated in many human cancers. So, again, just to kind of summarize this information about microRNA. So you need microRNA. You do have lots of microRNA, 1,000 to 200,000 individual microRNA molecules in the human cells. And you use this microRNA actually to stop the synthesis of protein, right? To stop synthesis of protein. Now, what happens if you don't have enough of this microRNA? Well, then you overproduce the protein that this microRNA is regulates, and this can lead to a cancer development. And actually, yes, in many human cancer, we have abnormal expression of microRNA genes. So we have either too many or too few of this microRNA. Okay, Another way to regulate the gene expression are microproteins. Microproteins are fewer than 100 amino acids long. They can fit inside nooks and crannies of large proteins, such as cell surface receptors and ion channels, affecting their function. So those microproteins, they can sit on the top of normal protein, and it can change their function. The genes that encode microprotein may lie within other genes, but the microproteins are not just part of larger proteins. One type of microprotein is like a garbage disposal, mopping up messenger RNA molecules that are no longer needed. Right, so if we let's just go back to the slide where we mention all of these factors that affect gene expression, right? Control of gene expression. That means what genes will be on and what genes will be off. Because you cannot have all your genes working constantly all the time, right? Different cells might have different genes that switched on and other genes need to be off. How to control it? Well, that's what epigenetic is. So you can control the expression of genes by chromatin remodeling, right? That's what we said. We're gonna attach these different chemicals to DNA or to histones. We can regulate it by microRNAs, and microRNAs will bind to messenger RNA and turn off the translation of protein, or we can control it via microproteins. So microproteins will attach themselves to other proteins and they can control their function, make them over-functioning or under-functioning, right? So that's all epigenetic control of gene expression. Okay, so now where did we stop? So here are microproteins, so here's our next slide. So only 1.5% of human DNA encodes proteins. Rest of genome includes, so if you look at your DNA, right, isn't it amazing? 1.5% of this DNA has instruction how to make protein. Then what is the rest of DNA? Well, the rest of genome include viral DNA, non-coding RNAs, introns. Introns are part of DNA that do not code for proteins, promoters and other control sequences. So part of DNA that doesn't make proteins, but important in a control of protein synthesis, and repeated sequences. Most of the genome is transcribed, however. A DNA sequence is not just junk. So all of this is not junk as we used to think, because we used to think that, well, this 1.5% of DNA encodes for protein, and the function of your DNA is to carry information about protein synthesis, right? And this is all like a junk DNA. Of course, it cannot be true. You cannot have more than 98% of your DNA that is just junk, right? We just either we don't know what this protein, DNA does, right? And or we didn't discover a function of this DNA yet. About 8% of our genome is derived from RNA viruses called retrovirus. So the 8% of your DNA has a DNA of a virus that is built inside your DNA. It's an evidence of past infections. Sequencing tends to increase over time, and the genetic material in the human chromosomes are called human endogenous retrovirus, or HERVs. So you have DNA of your virus in your own DNA, right? And it's called endogenous retrovirus, or HERVs. Most HERV sequences have exchanged part, recombined, and mutated to the extent they no longer make us sick. right? This DNA of the viruses cannot make us sick because it mutated too much. Intriguingly, people with certain diseases like AML, that is acute myeloid leukemia, multiple sclerosis, melanoma, they have overexpressed of HERVs. So it's interesting if this is a part of viruses in our DNA, and it doesn't make us sick, but at the same time, in certain diseases, we have overexpressed of this material. So is it then related to those diseases? This is something that we still need to find out. Nearly all of our genome can be transcribed. That means we can make messenger RNA, and much of it is in the form of non-coding RNA. So we will make a copy, right? Transcribe means we're gonna make a copy. If we make a messenger RNA, right, that's gonna be coding RNA. If we make ncRNA, those are non-coding RNA. That means they do not code for proteins. So this non-coding RNA includes all RNA other than messenger RNA. So all the RNA that your cells make from DNA rather than mRNA, called ncRNA. For example, tRNAs, that is transfer RNA. Or our 500 or so types of tRNA genes accounts for 0.1% of our genome. rRNAs, ribosomal RNAs, 243 types of rRNA genes are grouped on 6 chromosomes. Hundreds of thousands of other ncRNAs exist. They transcribe from pseudogenes, and they are not translated into proteins, right? So everything that is not that is transcribed, right? When the part of DNA genome that is transcribed, so we make RNA from DNA. That's what transcription is. But if they do not code for protein, we call it NC, non-coding RNAs. Now they can be transcribed from pseudogenes. They are not translated into proteins. So it can be ribosomal RNAs that are part of ribosome, transport RNAs that needed for protein synthesis, but these tRNAs themselves not gonna be translated into protein. Another class of RNA molecules are the 12,000 long non-coding RNAs. We have 200 or more 200 or more nucleotides make these RNAs. They are transcribed from exons, introns, and regions between genes associated with chromatin and control gene expression. So lots of the RNA molecules that are transcribed from DNA, right? But they do not code for proteins, they for control mechanisms. A third of the long non-coding RNAs are found only in primates and mainly only in brain. So we have this long non-coding RNAs, So what they are for? If we cannot make proteins from them, well, that's for control. And a third of them are only found in primates in the brain. So maybe it's a clue how and what makes us human. So if we don't understand completely what those RNase molecules are for, doesn't mean that we don't need them. Obviously, we need them, and especially if most of them found in our brain, so maybe they that's what make us who we are, right? Okay, so human genomes are riddled with highly repetitive sequences that hold that hold a different type of information than a protein's amino acid sequence, and this is called repeats. Actually, we have lots of repeats in our DNA. Transposons are the most abundant type of repeats. Transposons are sequences that jump about the genome. So they jump from one part of DNA to another part of DNA. sometimes creating or reversing mutations and altering the cell's genetic identity and genome size. Rare classes of repeats are telomeres, centromeres, and copies of pseudogenes. So, lots of our DNA is made of repeats. So, it's the same segments over and over and over again, right? Some of these repeats, simple sequence repeats, or another name is short tandem repeats, STRs. Those are repeat DNA unique to each person. And you inherit your STRs from each parent. And this is what used for DNA fingerprinting. So you probably heard about, well, if crime was committed, for example, or if you wanna find who is the father of a child, we use DNA testing, analysis of DNA. And we say like, well, it's 99% accurate, right? So if somebody left their DNA, we can find a person who committed this crime, for example. But did you think about, well, how it is possible? Humans have very similar DNA. We all have the same DNA. Even if you take human DNA, right, and compare it to chimpanzee DNA, you probably read somewhere we are like 98% similarities. Like if my DNA is 98% similar to chimpanzee DNA, how they can be sure, I committed a crime? But those similarities are genes that code for proteins. So we have 99.9% among humans, all these genes that code for proteins, they are the same. But what we have unique are those repeats. Those repeats do not code for protein, right? So for example, in this person, we have AGAT and he has 7 repeats. So AGAT 7 times. This person have AGAT only 4 repeats. This person have AGAT 12 repeats. And back to this person, okay, then we have another AGAT that 6 repeats, 19 repeats, 14 repeats, right? CTR1, CTR2. Now, those repeats of DNA do not code for protein, and those are the ones that you need. And that's what we use STRs, short tandem repeats, to have our DNA fingerprinting. This is how we identify people. Because if genes are the same, those repeats are unique for each individual. And you pass those repeats to your kids. Pseudogenes is a segment of DNA that structurally resembles a gene, but is not capable of coding for a protein. Most often derived from genes that have lost their protein coding ability due to accumulated mutations that have occurred over the course of evolution. Pseudogenes were once called genomic fossils and treated as junk DNA several years. Nevertheless, it has been recognized that some pseudogenes play essential roles in gene regulation of other parent genes, and many pseudogenes are transcribed into RNA. So some of our genomes is made of pseudogenes. Pseudo means false. So here's for example, common defect of pseudogenes. We have like missing promoter. So if you don't have a promoter, then we cannot make RNA copy of this gene. We cannot transcribe it. Or we can have a missing start codon, or we have some mutations within, or partial deletion. So now we have part of DNA that cannot be transcribed and translated into protein, right? So that's what our pseudogenes are. And you can see on this a diagram that what our genome is. So that's our entire genome, all our DNA. Only 1.5 are protein coding genes. 26% are introns. Introns are not transcribed. They can be transcribed, they cannot be translated into protein. We have transposons, 45%, that is a part of DNA that jump from one part of DNA to another part of DNA. We have simple sequence repeats that we just described that make your fingerprinting, 3%. Segmental modification, viral DNA, 8%. Then about 8% we have telomeres, centromeres, and other dark-stained DNA, heterochromatin. That is a part of chromosome, but again, it's not code for protein. And about 3-5% of other that we, like for different functions probably. But this is amazing how so large amount of your genome does not code for proteins actually. Right, so, and this diagram, we're not going over all of this type of sequence of your DNA and functional characteristic, but we just look at some. For example, viral DNA. So part of your DNA is viral DNA, about 8%, right? That's evidence of past infections. tRNA, rRNA, those are part of the rRNA, part of the ribosomes, and tRNA important in protein synthesis. They're gonna transfer amino acid to a messenger RNA to make proteins. So here's our pseudogenes. DNA sequences very similar to known genes that are not translated. On another one, we have our telomeres, centromeres. That telomeres, they protect chromosome tips. Centromeres found in inside the chromosome, and that's where two daughter chromatids are attached. And it's attachment of spindle fibers when cells divide. Right, so introns, part of the genes that are cut out of messenger RNA. We have promoters and other control sequences. That's a part of your DNA that does not code for protein. A microRNA that we already discussed that control translation of many genes. Okay, so that's it for this chapter. So I just want to remind you that epigenetics and gene expressions that we discussed in this chapter is about those chemicals that modify what genes are working and what genes inside your cells do not work, right? So epigenetic factors are chemicals that attach themselves to your DNA, to your histones of DNA, to messenger RNA, to maybe other proteins, and it might be small proteins attached to bigger proteins that regulate the expression of DNA molecules and function of the cells without altering DNA directly. And this is what you call nurture. So you have nature and nurture. So nature is what your genes are, and nurture are those chemicals that you can alter through your diet, through your lifestyle, through some bad habits like smoking or drinking. So you can increase and decrease the amount of epigenetic factors, those chemicals that will affect your DNA expression, right? Okay, so that's it for this lecture. Thank you for watching, and I hope it was helpful.